discussed, it is defined as number of grams of iodine absorbed by 100 gram of fat or oil. So iodine number is nil in the case of saturated fatty acids, whereas iodine number is high or more in the case of polyunsaturated fatty acids when compared to monounsaturated fatty acids. So iodine number increases with increasing the number of double bond. That means it depends on degree of unsaturation. Iodine number, it depends on degree of unsaturation. So those points we can highlight. So by determining the iodine number, we come to know the position of double bond, where double bond exactly located. So it helps in determining the position of double bond, and so it also signifies by determining the iodine number. So we come to know that, so whether the quality is high. Okay, so the quality of oil sample is high, the quality of the lipid sample is high. So there will be the normal range, iodine normal range for the oils, for the cooking oils. If the value exceeds, then the quality, so we come to know the quality. So what is the quality of the respective oil sample? So there should be a nominal range of iodine values for the cooking oils. So by determining the oils, we come to know whether the iodine value is fine or whether the iodine value is very low. So that means indirectly, so it states that the quality of oil is not that much good. Okay, so all these are chemical tests, so which includes acid value or acid number, saponification value or saponification number, and the iodine value and the iodine number, which helps in determining the composition, determining the quality, and determining the purity of the fat or the lipid. Okay, so these are very, very important. Okay, so overall it helps in characterizing the fats. It helps in characterization of oil or the fats. So that is the very important point that so we need to understand. So by knowing these values. Okay, so because the physical and chemical property is over, like characterization is over. So moving on to classification of lipids. Lipid classification. So particularly we are going to concentrate on compound lipids. So we know that lipids are classified. So here simple lipids based on composition, simple lipids, and compound lipids. Then derived lipids. So by definition, so we come to know what is the difference between simple, compound as well as derived lipids. So here simple lipids, they are esters of fatty acid and alcohol. Esters of fatty acid and alcohol. So that is what we call it as simple lipid. Whereas when you talk about compound lipid, when you consider compound lipid, they are esters of fatty acid, and along with fatty acid contains alcohol. So maybe glycerol or other alcohol, right? And apart from that, it contains additional chemical groups. So when it contains additional chemical groups apart from esters of fatty acid and alcohol, so we consider it as compound lipid. Whereas derived lipids, they are derivatives which are obtained from simple and compound lipids. They are considered to be derivatives obtained by hydrolysis. Derivatives which are obtained by hydrolysis. So these are the major difference. So from the definition we can understand or we can differentiate what is simple, compound, and derived lipids. Simple lipids, esters of fatty acid and alcohol. Compound lipids, esters of fatty acid and alcohol. In addition, it contains that means glycerol. Okay. So here the derived lipid, it mainly consists of derivatives. They are derivatives of lipids which are obtained by hydrolysis of compound lipids. Okay, they are derivatives which are obtained by hydrolysis. Okay, so they are simple compounds which are obtained from either compound lipids or simple lipids. So we don't want to discuss much about simple lipids as well as derived lipids, but we should know. So how it is further classified? How this simple lipid is further classified? And we need to know the example of simple lipid. So only that much is enough here. So simple lipids, again here it is categorized into fats, neutral fats, we call it as neutral fats, and then waxes. So I'll tell you the basic difference between the neutral fat and the waxes, and the example as well. So neutral fats, we can give example TAG, triacylglycerol, TAG. So we can give example TAG. What is neutral fats? Neutral fats, they are present in large quantities when compared to compound lipids and derived lipids. The distribution is more. Neutral fats, so they are abundantly present, the distribution is more, and it is made of, that is glycerol, one molecule of glycerol and 3 molecules of fatty acid. Okay, so that is what is triacylglycerol. So it is also called as triacylglycerol. So triacylglycerol is also called as, that means neutral fats. We know the structure of triacylglycerol or the neutral fats. They serve as padding material. It serves as insulating material. Okay, and we have discussed about the physical properties of fats or lipids. Okay, so even the characterization also, iodine number, acid number, and the saponification number and all those things. So triglyceride or nothing but reserve storage, reserve form of the lipid, reserve form of the fats. They are the storage form of fat or lipid, mainly stored in adipose tissues, and they serve as insulating material, they serve as padding material which protect the vital organs from the mechanical shock or the damage. Okay, so that is the ultimate function. So of these neutral fats, familiar example is triacylglycerol. But when you consider waxes, they are esters of fatty acid, and along with that it contains other alcohols. So mainly cholesterol esters, or we call it as, there are different type of waxes. So true waxes, cholesterol waxes or esters, and vitamin A and vitamin D esters or waxes. So these are some common examples for the waxes. So they are esters of fatty acid and the alcohol. Other than glycerol, it contains alcohol other than glycerol. Okay, so true waxes, for example, if you consider true waxes, so where it consists of fatty acid, along with that it contains higher alcohol, acetyl alcohol. So when it contains higher alcohol apart from glycerol, so that is considered as true waxes. Cholesterol esters, apart from cholesterol is esterified with fatty acid. So we call it as cholesterol esters or cholesterol waxes. Similarly, vitamin A and vitamin D, they are esters of palmitic and stearic acid. Okay, so all these comes under waxes. So that is about simple lipid, neutral fat, and waxes. So two categories were there. Example for these waxes: true waxes, cholesterol, that means esters or waxes, and then vitamin A or vitamin D, esters or waxes. Now moving on to compound lipids. So first, I'll just explain how it is further classified. Then we will move on in detail about each compound lipids. Okay, so we know what are compound lipids. So compound lipids are the compounds which consist of fatty acid, then alcohol, probably glycerol, and then along with that, in addition, it contains chemical groups, additional chemical groups in its structure. So all together it forms the structure of compound lipids. So when we say compound lipids, so what are the major compound lipid fractions is that? So one is phospholipid, I'll write it here. So phospholipids, and other major compound lipid is considered to be glycolipids, glycolipids, and the other one is here lipoproteins. Lipoproteins. So we'll discuss later. So once we finish the classification, entire classification, then we'll go in detail about each compound lipid, their structure and the functions. Again here phospholipid, again this classified based on type of alcohol, based on the type of alcohol group. So this phospholipid is categorized into phosphatides, and here this one is phosphoinositides, and third category is phosphosphingosides. So based on type of alcoholic group, so these phospholipids are further classified into glycerophosphatides, phosphoinositides, phosphosphingosides. Similarly, glycolipids again two different categories. One is cerebrosides. The other one is gangliosides. Lipoproteins. So here, so based on the electrophoretic mobility, okay. So they are categorized as VLDL, very low density lipoprotein. Then that is high density lipoprotein and chylomicrons. I think you are able to see here, right? So lipoproteins are categorized as chylomicrons, VLDL, LDL, and HDL, high density lipoproteins. So coming to derived lipids, I'll just finish the classification charts. Then we will move on to in detail about the respective compound lipids and their structure and their functions, biological importance. So we'll just finish this classification chart because it's very, very important. And here, considering the derived lipids, we know they are derivatives which are obtained from simple or compound lipid on hydrolysis. So derived lipids, it includes three different categories. That means fatty acid. One is fatty acid. So here it includes fatty acid, and this one is glycerol. And under fatty acid, so we have three different types: saturated, unsaturated, and cyclic fatty acids. Saturated, unsaturated, and cyclic fatty acids. Okay. So this classification chart is very, very important. So when they ask classification, and here in our syllabus we have detailed notes on compound lipids, but then if they ask general classification of lipids, so you need to draw this flow diagram. This is very important. So wherein we can see the further classification. So of each and every lipid fraction, simple lipid, how it is classified further, and neutral fats, and then here waxes. Similarly, there are compound lipids. So under compound lipids, what are the different compound lipids? That includes phospholipid, and then glycolipid, and then so there are lipoproteins and their subcategories. Just mention. So mention the names. Okay, no need to give example. Just mention the sub-classification. Okay, that is what I meant here, right? That is very important. Then coming to the derived lipids, again here in derived lipids, they are derivatives, and mainly when fat is being hydrolyzed, so it is hydrolyzed to fatty acid and glycerol. So we need to remember that. So there are two categories. One is fatty acid and the other one is glycerol. So under fatty acid, so different type of fatty acids are there: saturated fatty acid, unsaturated fatty acid, and then cyclic fatty acids. So we need to know the difference. What is saturated? What are the characteristic features of saturated? What are lower saturated? What are higher saturated fatty acids? Example. And then similarly unsaturated. Okay. Example. Then cyclic fatty acids. So now we can start with the classification and the detailed classification of compound lipids. I just mentioned compound lipid. What are compound lipids? Compound lipids are esters of fatty acid and alcohol, and in addition it contains chemical groups. So that chemical group may be phosphoric acid, phosphoric group. It may contain phosphoric group or phosphoric acid, or it may contain some nitrogenous base, choline, or it may contain other nitrogenous base, ethanol, amine. So it differs with the nitrogenous base as well. Okay. So that additional chemical group includes, that is phosphoric acid or phosphate group and nitrogenous base. It differs with nitrogenous base as well. Okay. So those are two which are considered as chemical groups. So in addition to chemical groups, compound lipids have fatty acids as well as, that means, alcohol, that is glycerol. So all together it constitutes the structure of compound lipid. So let us see the structure of compound lipid one by one. So we go with the phospholipids. So phospholipid is very, very important. So we go with the phospholipid, and so here the structure of phospholipid. So we need to know the structure and classification, how phospholipids are further classified. Okay. So phospholipids, when you say phospholipids, so first we'll discuss what is the importance of phospholipid, or where phospholipid is mainly distributed. Phospholipids, it mainly increases the rate of fatty acid oxidation. All these are general functions, so which are applicable for even glycerophospholipids, glycerophosphatides, phosphoinositides, and phosphosphingosides. So phospholipids, where it is distributed, the distribution of phospholipid, we know that. So they are mainly present, and their location is mainly targeted on cell membrane. So plasma membrane of eukaryotic cells are rich in phospholipids, glycolipids, and other lipid fractions. It involves even cholesterol as well. So they are the part of the cell membrane. So they are the usual constituents of the cell membrane. So phospholipids are the usual constituents of the cell membrane. Which cell membrane? Cell membrane, so mitochondrial membrane, myelin sheaths, even microsomes. So all these cell membranes are considered to be rich source of phospholipids. So the cell membrane of eukaryotic cells, plasma membrane, rich in phospholipids. So that is the main distribution of phospholipid, where phospholipid exists. So what is the role of phospholipid? So we can categorize phospholipids into different types. But then what is the role of phospholipid is that phospholipids, it increases the rate of fatty acid oxidation. So what happens when fatty acid is oxidized? When fats are hydrolyzed, we get fatty acid and the glycerol. When fatty acid are simple compounds, the simple lipids, when it is being oxidized, and fatty acid is oxidized, ultimately it increases the concentration of acetyl-CoA. It is converted to acetyl-CoA. When it is converted to acetyl-CoA, then acetyl-CoA enters the PCA cycle, and then it is completely oxidized. That means it releases energy. Okay, so the energy, the oxidation of fatty acid, it provides energy, right? So that results due to increased fatty acid oxidation. So when fatty acid is getting oxidized, when more amount of fatty acid is getting oxidized, it increases overall the concentration of acetyl-CoA, and instead of only limited acetyl-CoA can enter the PCA cycle. If the concentration exceeds beyond the limit, so that time it is diverted to other synthesis, like it may stimulate cholesterol biosynthesis, or it may be involved in ketone body biosynthesis. Okay, so it is involved in some other biosynthesis. But normally, when fatty acid is oxidized, ultimately it is converted to acetyl-CoA that enters PCA cycle, which is completely oxidized. So that is a normal process which provides energy. Like how carbohydrates, how proteins provide energy. Similarly here, fatty acids, when it is getting oxidized, oxidized, ultimately it provides energy only when it is converted to acetyl-CoA and getting oxidized via this tricarboxylic acid cycle. So phospholipid, it increases the rate of fatty acid oxidation. So it stimulates the oxidation of fatty acids. So it promotes the fatty acid oxidation. That means it provides energy. Indirectly, it provides energy by stimulating the fatty acid oxidation. Then, so these phospholipids which serve as prosthetic group for certain enzymes. They serve as prosthetic group for certain enzymes, non-protein substances. They serve as prosthetic group for certain enzymes. So it is considered to be, it is a constituent of blood grouping substance. They are associated with the blood grouping substance. Then it enhances or stimulates the transport of inorganic ions. That means some of the phospholipids serve as carrier ions. It serves as carrier. So for transporting the inorganic ions across the cell membrane. So it is involved in transporting ions across the cell membrane. It facilitates the transport of ions across the cell membrane. That means it serves as a carrier molecule. So it serves as a carrier molecule, right? So, and it is the normal constituent. We know that these are normal constituents of cell membranes: mitochondria, microsomes, myelin sheath, plasma membrane, eukaryotic plasma membrane are rich in phospholipids, apart from glycolipids and other lipid fractions. So it is considered to be one of the major lipid constituents that constitute the cell membranes. Okay. So all these are important functions. But when you consider the classification, so I just mentioned the classification of phospholipids. Further classification of phospholipids based on type of alcohol. So when it contains especially glycerol as alcohol, so it is named as glycerophosphatide. We'll go one by one so that it will be clear, right? So first we'll finish glycerophosphatides. What are glycerophosphatides? What are the functions of glycerophosphatides? So glycerophosphatide means it is a phospholipid, and here it contains glycerol. So here the alcohol, here the type of alcohol, considering this glycerophosphatide, so the type of alcohol is considered to be glycerol. That's why it is named as glycerophosphatide, right? So based on type of alcohol, so type of alcohol, so type of alcohol, so phospholipids are categorized into three different categories. Under that, first one is glycerophosphatide. So example we can discuss. First we can see the example. So first I'll write one by one. So the first structure is cephalin. Okay. So it is cephalin. Example is cephalin. Glycerophosphatide. Example is cephalin. So what is the cephalin? What is the chemical structure of cephalin? What it constitutes of? So it consists of, so CH2O, C11O, R1. CH2O, C11O, R2. Then CH2O, phosphate is esterified. I said phosphate is esterified. CH2, CH2, NH3 plus. Okay. So this is structure of cephalin. So cephalin is a glycerophosphatide. Glycerophosphatide, it is a phospholipid. Where glycerol is considered to be alcohol. It contains one molecule of glycerol. So this is glycerol, right? And these two are fatty acids. So this is fatty acid, and this one is phosphate. That is chemical group. Chemical group is phosphate group, and then it contains nitrogenous base. So this nitrogenous base, here it is ethanol amine. This one is ethanol amine, right? And this one is phosphate. This is phosphate, and this one is glycerol, right? And this is fatty acid. Okay, so all together it forms structure of cephalin. Okay, so here we should remember what is compound lipid. Compound lipid is they are esters of fatty acid, glycerol, and additional chemical groups. Esters of fatty acid, glycerol, and then additional chemical group. One is phosphate, and the other one is nitrogenous base. So when nitrogenous base is ethanol amine, CH2, CH2, NH3 plus ethanol amine, then it is named as cephalin. Okay, so then it is named as cephalin. So what is cephalin? Cephalin is a phospholipid, and mainly it contains ethanol amine as nitrogenous base. So the other name of cephalin, so how can we write the other name of cephalin is that we can write it as phosphatidyl ethanol amine. Ethanol amine. So we write as phosphatidyl ethanol amine. Okay, so cephalin is also called as phosphatidyl ethanol amine, right? So we need to be clear with the structure because these structures are very, very important, especially compound lipids. We have to draw the structure of compound lipid, each and every compound lipid. For example, phosphatidyl ethanol amine. They might ask, write down the structure of lecithin, write down the structure and function of, that means phosphatidyl serine or phosphatidyl plasmalogen or phosphoric acid. So we need to familiarize with the structure of individual compound lipids, individual phospholipids, and their functions. So we need to know the functions, generalize the function of the phospholipid as well as specific function of the phospholipids. For example, if it is lecithin, let us say lecithin, right? So I think this structure is fine. You will be able to understand this structure. So it contains fatty acid, glycerol, phosphate group or phosphoric acid, and then nitrogenous base. Nitrogenous base is ethanol amine. I have given the structure in the LMS content as well. So please go through the structures and please go through the videos. Okay. So it will be very helpful. So you need to practice these structures. That is very important. So now we know the structure of cephalin. So how can we write the structure of lecithin? So what is the structural difference between cephalin and lecithin? So we can easily differentiate the structure of cephalin and lecithin. So simply by substituting the nitrogenous base. So it differs with nitrogenous base. Rest all it is same. It contains fatty acid, it contains glycerol, it has glycerol, and it has phosphoric acid, but it differs with the nitrogenous base. So what is the nitrogenous base in case of lecithin? So that we need to understand. Okay. So in case of lecithin, the nitrogenous base is, so instead of ethanol amine, so it contains, that means choline, 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 right? So CH2, and so CH3 reacts choline. So choline is a nitrogenous base. So instead of ethanol amine, so now this is not cephalin. So now this has to be named as, that means lecithin. Lecithin, lecithin. And this lecithin, so if the structure is lecithin, so instead of ethanol amine, so we should write it as, we should name it as phosphatidyl choline. Okay. So instead of ethanol amine, here the nitrogenous base is choline. So the nitrogenous base here is choline. It is replaced with choline. So it is named as phosphatidyl choline, and the other name is lecithin. Similarly here, lecithin function, the function of lecithin we have to highlight. What is the role of cephalin? So both lecithin and the cephalin, so mainly apart from the generalized functions, whatever I mentioned, like fatty acid, it increases the rate of fatty acid oxidation. They serve as blood grouping substance. All that points we have discussed. No, apart from that, it serves as transport. That means carrier molecules, promoting the transport of inorganic ions across the cell membrane. So all these points we had discussed. Constituent of cell membranes. So all these points we had discussed. Apart from that, specialized functions of cephalin and lecithin. So those two facilitates the formation of lipoproteins. It facilitates the formation. Lecithin and cephalin, so it is involved in formation of lipoproteins. It facilitates the combination of lipids with proteins, so that it is involved in synthesis of lipoproteins. It facilitates the formation of lipoproteins. Different types of lipoproteins are there: LDL, VLDL, HDL. So it facilitates the formation. That is one major function of lecithin and cephalin. And especially when you consider lecithin, we know that here the nitrogenous base is choline, right? So choline is a nitrogenous base. When it combines with acetyl-CoA, like acetyl-CoA, it is acidified with acetyl-CoA, it forms acetyl-choline. So we know what is the role of acetyl-choline. Acetyl-choline is a potent neurotransmitter. It facilitates the transmission of nerve impulse, especially in the synapse region, synaptic region, right? Synapse. It facilitates the transmission of nerve impulse. So those functions we can highlight. And the cephalin, it also prevents the fatty liver formation. It prevents the fatty liver formation. Okay. So fatty liver formation can be prevented by the action of this lecithin and cephalin. And moreover, this lecithin lowers the surface tension of lung alveoli. So lung alveoli, inside the lungs we have the smaller units called as alveoli, alveolar membrane. So particularly the surface tension of alveoli is mainly due to the presence of this lecithin. And this lecithin, it lowers the surface tension of lung alveoli. In some cases, like premature, that is infants, so where the lung alveoli is not properly developed. So, if it connects with the phosphatidyl-choline, that means lecithin, so ultimately it results with the distress syndrome, breathing trouble, respiratory distress. We call it as respiratory distress. That means syndrome. Okay. So it lowers the surface tension, and the role of phosphatidyl, that is choline, and the phosphatidyl ethanol it lowers the surface tension, particularly of water in the small intestine. So thereby it promotes the emulsification process. Whenever the surface tension of water is lowered, so that enzyme action will be more. So it stimulates the action of intestinal enzymes. So where the enzyme secretions reaches the target region, so thereby, so it promotes the emulsification process. So the breakdown of lipids, complex lipids into smaller lipids, and thereby it promotes the digestion. It promotes the hydrolysis of fats, so that it is converted to fatty acid and glycerol. So all these functions we can highlight, specific functions. That is what I meant. So we need to highlight the specific functions of these. That means phospholipids. So we need to know the difference. Okay. So under glycerophosphatide, under phospholipid, today we are discussing about glycerophosphatide. Under glycerophosphatide, so we have different types. The first one is cephalin, and the second one is lecithin. So we need to know the structural difference between the lecithin and cephalin. Okay. So first one is lecithin. The other name we call it as phosphatidyl ethanol amine. So there the nitrogenous base is ethanol amine. Whereas in the case of lecithin, so instead of ethanol amine, so it is substituted with choline. Okay, the nitrogenous base is substituted with choline, so it is named as phosphatidyl choline or lecithin. Okay, so we need to know the structural difference as well as the specific role played by the phospholipids. Then we have phosphatidyl serine, then plasmalogen, and phosphatidic acid. So all the five, so that comes under glycerophosphatides. Okay, so all the 5 which are considered to be glycerophosphatides. For example, if you take phosphoinositides, it serves as a second messenger, okay, for calcium regulated hormones. So hormonal action. Like that, each and every phospholipid, so they have their own role. Okay, so apart from the generalized functions of the phospholipids. So those things are very, very important. If the specific phospholipid concentration is increased or when it gets deposited, so it leads to the even disorders. It results with clinical conditions, clinical disorders. Probably we can discuss that as well. So the associated with the phospholipid structure, functions, and the associated disorders which are related to different type of phospholipids. So that is very, very important. So today we just started classification. Under classification chart, we discussed what are simple lipids, what are compound lipids, what are derived lipids, the basic differences and their sub-classification, and then we started with so especially the glycerophosphatides. So how based on type of alcohol, how phospholipids are categorized. So 3 categories: glycerophosphatides, phosphoinositides, and phosphosphingosides. So under glycerophosphatides, we finished 2 structures. One is cephalin, the other one is lecithin. So 3 more structures are left under the glycerophosphatides. Then afterwards we move on to phosphoinositides and as well as phosphosphingosides with specific example and their functions and their roles. Okay. So with this, today's lecture comes to end. Students, if you have any doubt, any queries, so you can raise your queries and kindly go through the LMS contents. So LMS contents. So for these 2 lectures were open. Okay. And some of them messaged me that we are unable to retake those lessons. So that we will check and probably we can do the needful so that we can repeatedly take those lessons as well. Okay, so pertaining to these topics. So probably this classification we can continue in the next lecture as well. Okay, so phospholipid, it is a very important part, especially in the classification alone. Phospholipid alone is very, very important. Okay. Then we'll discuss glycolipids and then lipoproteins. Then probably we can go to the derived lipids, different types of fatty acids, and then we'll talk about the glycerol. Okay. So and then we'll move on to the other topics like prostaglandin, and we have membrane models. Okay. So all those things are there. Okay. So if you have any doubts, students, you can ask. Otherwise, we'll wind up the session. So is it clear, students?
Yes, sir.
Can we end the session? I think regarding the assignments, okay, your internal assignments, probably within 1 or 2 days, I'll just discuss with Sriram sir. We'll let you know the topics for the internal assignments, that is course assignments, especially for biochemistry 5. So with the dates, okay. So if there are no more queries, so we can end the session.